I came through the training program here and started my lab at Brigham Dana-Farber about 2 years ago. And this is a story that's currently in press at Cell Reports. It's the first story for the lab, so it's a proud moment. My lab is focused on myeloid malignancies and especially on myeloproliferative neoplasms, which, as you probably all know, are clonal disorders of hematopoiesis that arise in the hematopoietic stem cell compartment. And they are named for the phenotypes of the disease, polycythemia vera, essential thrombocythemia, and myelofibrosis. But as we all know, based on work, this had been sort of hypothesized for a long time and was definitively demonstrated in 2005 that these diseases have a common molecular basis. And the most common molecular abnormality is the inactivating mutation in Jak2 kinase, which was identified in 2005 in almost all patients with polycythemia vera and about 50% of those with essential thrombocythemia and myelofibrosis. And the story I'm going to tell you today pertains to this mutation. A small number of patients have activating mutations in the thrombopoietin receptor MPL, and then most recently, the majority of the remaining patients have mutations, recurrent mutations in an endoplasmic reticulin chaperone protein called calreticulin. And very interestingly, these mutations tend to be mutually exclusive and not entirely clear how calreticulin induces MPN. That's the focus of work in my lab and many other labs. But as I said, today I'm going to focus on Jak2V617F. When I was a postdoc with Gary Gilliland and Ben Ebert, we developed a mouse model of the Jak2V617F mutation. This is a conditional knock-in mouse model where the mutation is expressed from the endogenous murine Jak2 promoter. And essentially, these mice get a phenotype very reminiscent of the human disease polycythemia vera. And they develop marked erythrocytosis, splenomegaly, and very prominent extramedullary hematopoiesis. They have expanded committed erythroid precursor cells in the spleen, as you can see here. These are CD71, TER119 double positive cells. They demonstrate leukocytosis. They have marked megakaryocytic abnormalities, and they develop transformation to reticulin fibrosis with long latency, analogous to the fact that typically patients with PV who develop fibrotic transformation do so after decades of having PV. So we think this is a nice model and recapitulates the human disease very closely. And so when we developed the model, one of the first things we did, of course, was to use it as a preclinical model to study various therapeutics. And of course, at that time, there were many drug companies developing kinase inhibitors to Jak2. And we worked with several companies, including with this small company, Targaden, who developed a selective Jak2 inhibitor, TG101-348. This drug was subsequently bought by Sanofi, and unfortunately was taken out of late phase clinical trials due to excessive neurotoxicity. However, we found this drug to be very clinically effective in mice, and we found that the mice had significant reductions in splenomegaly and also reductions in their elevated blood counts. And this, of course, was subsequently found in the clinical trials. This is the COMFORT trial, which compared ruxolitinib, which is the current only FDA-approved JAK inhibitor. It's approved for myelofibrosis and PV patients that are resistant to hydrea. And as you can see here, patients who are treated with ruxolitinib as compared with placebo develop significant reductions in splenomegaly. These occur early and are generally sustained and occur in the majority of patients. But what we found, and I'm not going to show you this data in the interest of time, but we tried to address this question of whether we could eliminate the disease propagating stem cell population in the model, and we defined that this population was contained in the long-term stem cell compartment. And so we did serial transplantation experiments where we would treat the mice with inhibitors and then try to transplant the disease in the face of drug inhibition. And we were not able to eradicate the stem cell propagating compartment, and this indicated to us that the stem cell compartment was insensitive to treatment with the drug. And again, this perhaps has been one of the big disappointments of the JAK inhibitors in that similar findings have been seen in the clinical trials. and they have been demonstrated not to be clonally selective in the same way that imatinib is for CML disease. So it's in marked contrast to the efficacy of imatinib at reducing BCR-ABL transcript levels very rapidly. So this is the data, the Jak2V617F allelic burden data from the COMFORT-1 study. There were 150 patients who received ruxolitinib, and as you can see, 100 of those patients, after 24 weeks of treatment, had a median reduction in their Jak2V617F allelic burden of just 11%. And in the COMFORT-2 study, which compared ruxolitinib to best available therapy, which was hydroxyurea in the majority of patients, the median reduction was just 8% after 72 weeks of treatment. So we were motivated to try to understand if there were differential molecular dependencies of JAK2 mutant cells and novel targets that we could identify to try to improve on these sort of non-selective results of the JAK2 inhibitors. And so to do that, we basically went back to a data set, a gene expression Affy-array data set that had been generated by Edwin Chen, who was a postdoc in my lab, and prior to this had worked with Tony Green at Cambridge University in the UK. And what Edwin and Tony had done was perform clonal analysis in MPN. And so they would take a blood sample from a patient and do an erythroid colony assay, and then actually pluck and genotype individual colonies for Jak2V617F, and then pool all the mutant ones and pool all the wild type ones and perform any downstream analysis that they wished to do. So this was sort of in the days before single cell, but still very powerful, and mostly they did gene expression on Affy-arrays and were able to compare wild type and mutant cells from within the same patient, but also mutant cells across the different phenotypes, disease phenotypes, EV versus PV versus MF. So we basically interrogated this data set to look for genes that were differentially expressed in JAK2 mutant cells as compared to wild type cells. And we were interested when we pulled out this gene, RegQL5, which we found to be differentially expressed in the JAK2 mutant cells as compared to the wild type cells across all the three disease phenotypes, PV, ET, and MF. And then I'm just showing you here, this is a qPCR validation of this finding in the ET and PV patients. So we found increased expression of RegQL5 in VF as compared to wild type cells. So this gene was interesting to us because of the fact that MPNs, as you probably know, are diseases that often remain very genomically and clinically stable for decades. Despite the fact that they're neoplasms and they're driven by oncogenes, and there's actually evidence of DNA damage accrual in these cells. And so we thought this would be an interesting gene to study to try to understand this conundrum of sort of DNA damage in these cells, but at the same time genomic and clinical stability. So RegQL5 is a member of the RegQ family of DNA helicases, of which there are 5. You may have heard of Bloom and Werner. There are some germline syndromes where patients have mutations in these and have cancer predisposition. And basically RegQL5 has this role in sort of as a guardian of the genome, and it localizes to the replication fork when the fork stalls, for example, if it encounters DNA damage. And therefore, it functions to stabilize the DNA replication fork and prevent its collapse and prevent the development of DNA double-stranded breaks. And its role in MPN biology had never been discovered, or had never been investigated, so we thought it'd be an interesting gene to study. And so essentially, this is the question we're trying to get at. As I said, there's work both in experimental models and also in primary patients to show that Jak2V617F can actually induce DNA replication stress and DNA damage. And we were interested in sort of looking at the pathways that might be relevant to counteract this, specifically in the context of replication stress. And this had been studied previously in the context of CML, but primarily looking at double-stranded break repair. So again, we thought this would be a novel sort of unexplored area for investigation. So the first thing we did was we generated cell lines from the mice. And actually, David Sykes here at Mass General, working with David Skadden, was kind enough to do this for us. And essentially, we took bone marrow from the mice and transformed them with estrogen-inducible HOXB8 retroviral vector. So these cells are transformed at the granulocyte macrophage stage of differentiation, and they're immortalized. And when you grow them in the presence of estradiol, HOXB8 is expressed, and so you can propagate these cells indefinitely. And so this gives you lots of cells to work with for protein studies, et cetera. And importantly, these cells demonstrated the pathognomonic feature of MPN, of Jak2 mutant MPN, which we see in patients, which of course is cytokine hypersensitivity. We also see this in the primary mice, and this is just demonstrated here. These are the cells grown under standard growth conditions. You can see differential phosphorylation of STAT5. So again, saying these are a relevant model to work with and for which to pursue studies. So we took the cells, and basically the first thing we did was, this is just showing the cytokine hypersensitivity. So if you grow the cells under standard growth conditions, 10% SCS, they grow at the same rate. If you grow them under 2%, the VF cells grow preferentially well due to this cytokine hypersensitivity. We went ahead and did a qPCR on these cells, looking at the RECQ family members, found increased expression of RECQ-L5 as we had seen in the primary patient samples, and interestingly did not see differential expression of other members of the RECQ family. So this seemed to be specific to RECQ-L5. Validated this using Western blot. Again, saw increased expression of RECQ-L5, but not of other family members. So then we're interested in how this pertained to Jak2. So we did various experiments knocking down and inhibiting Jak2. I'm just going to show you some of them here. This is where we took a human cell line. Set2 is a cell line that is heterozygous for the Jak2 mutation. HEL cells are homozygous for the Jak2 mutation. We treated them with INCB018424, which is ruxolitinib. And with increasing concentrations of ruxolitinib, you can see inhibition of STAT5 phosphorylation, as you would expect, but also a decrease in RECQ-L5 expression. When we knocked down RECQ-L5 using shRNAs, again, we saw decreased expression of RECQ-L5, but when we knocked down STAT1 or STAT5, we did not see any change in RECQ-L5 expression. And then we also looked at some other pathways. This is drug treatment again with PI3 kinase inhibitor and also with a MEK inhibitor. And we found that the PI3 kinase inhibitor resulted in decreased expression of RECQ-L5, but not the MEK inhibitor. And we did these studies with shRNAs, both in the mouse cell lines and in the human cell lines. And basically, in aggregate, the data shows that RECQ-L5 appears to be downstream of activated Jak2 PI3 kinase signaling. So we then wanted to sort of functionally assess the role of RECQ-L5, and so we did some proliferation assays, again with the cell lines from the mice, again using shRNAs against RECQ-L5. So when we grew the cells under standard growth conditions, this is 10% FCS, they grow equally well as I showed you before. If you knock down RECQ-L5, you see no differential effects on growth. However, if you grow them in low FCS here, this is 0.5% FCS, again you see that the VF cells grow preferentially due to activated Jak2 signaling and cytokine hypersensitivity. And now when you knock down RECQ-L5, we see this preferential effect on the VF cells. And so we think this recapitulates an endogenous replication stress by starving the cells, and that we can bring out this preferential dependence on RECQ-L5 in that context. And we can completely rescue this phenotype by just giving back nucleotides to the growth media, and then the growth will return to the levels of wild type. So essentially, as the title says, RECQ-L5 depletion sensitizes the V617F cells to endogenous replication stress. We then went on to interrogate exogenous replication stress using various pharmacological inducers of replication stress. And the obvious example to start with here was hydroxyurea, which is the most common treatment we use in the clinic for MPN, but of course is not preferential and inhibits wild-type and mutant cells equally. So again, we did our shRNA knockdown, looked at our proliferation assays, and in the context of treatment with hydroxyurea, we saw a preferential effect on VF cells as compared to wild-type cells. We also saw this for camptothecin, another known inducer of replication stress. And this effect is very specific to these drugs. We don't see it globally. For example, if we use DNA double-stranding breaking agents like etoposide or daunorubicin or mitomycin C, or we use oxidative stress inducers like hydrogen peroxide or picrolongamine, we don't see these effects. It's really specific to irinotecan, camptothecin, and hydroxyurea. Again, then, just to demonstrate that this was an on-target effect, we did a rescue experiment. So just to walk you through this, this is again looking at growth or viability of the cells in the presence of increasing concentrations of hydroxyurea. As you can see, when you knock down RECQ-L5, you get an impairment of the viability. If you rescue by overexpressing a cDNA for RECQ-L5, you don't see any rescue, basically because the shRNA is knocking down the cDNA. But then if you use a shRNA-resistant cDNA to rescue, so this is mutated at every third nucleotide, so the amino acid sequence is preserved, but the shRNA can't bind, you can see that you rescue back to the same as the VF cells. So this demonstrates that this is an on-target effect of RECQ-L5 knockdown. We've also tried to rescue with an enzymatically dead version of RECQ-L5, and that does not rescue, so it suggests that it's the enzymatic function of this helicase that is required for this function. So then finally we went to patients, and with the help of Dan DeAndrew at Dana-Farber, we were able to get some peripheral blood cells from patients. We took CD34 cells from their peripheral blood and then knocked down, these are myelofibrosis patients actually, knocked down RECQ-L5 and did our erythroid colony assay in the presence and absence of hydroxyurea, and then genotyped at the end to look for differential effects. And essentially, if you grow cells in hydrea, they make less erythroid colonies, and then if you knock down RECQ-L5, they grow even less well. And then if you deconvolute which cells are really contributing to this inhibition of growth, it's really primarily the V617F cells. So in primary samples, we also see this preferential sensitivity of the VF cells to RECQ-L5 knockdown in the context of hydroxyurea treatment. So then we went on to try to understand this mechanistically. So our model would be as such. Here's the DNA replication fork meeting a site of DNA damage. Replication fork stalling occurs. This would be the point at which RECQ-L would come in and allow the fork to recover and stabilize. In the absence of RECQ-L5, you would predict that the fork would collapse, and the end result of this would be double-stranded DNA breaks. So we basically assay this using what's called chromosome combing. And so this is actually a method where you can visualize the DNA replication tract and actually visualize individual DNA fibers that you label with BRDU analogs. So we did the first labeling stage with IDU, and then we did the second labeling phase in the presence of hydroxyurea with another BRDU analog, CIDU. And we did these for 20 minutes each. Then you harvest the cells, extract the DNA, and literally you stretch an individual DNA fiber on a glass slide and then stain with antibodies against IDU and CIDU. And essentially then you take pictures or visualize this with a microscope, and you see the DNA replication tract like such. And hydroxyurea obviously impairs DNA replication fork progression, and so you see shorter CIDU labeled pieces of DNA, and this would be indicative of increasing stalling with the shorter the green segment. So we went ahead then and did this in the absence and presence of RECQ-L5 knockdown, and this data is shown here. And essentially you can calculate the replication fork progress based on measurements of the length of these DNA fibers and converting this into a metric of fork progress. And so the first step, as I said, was done in the absence of hydrea, so we normalized that to 1 and then looked at progress in the presence of hydrea treatment. And as you can see, in general, there's less fork progress in the presence of hydrea treatment, but when you knock down RECQ-L5, you see that the VF cells are really significantly impaired in their fork progress. We did some other assays, but in the interest of time, I won't show you those. I'll just show you that we looked for double-stranded break formation using gamma H2AX foci, and we saw these increased gamma H2AX foci in the VF cells that were knocked down for RECQ-L5 in the presence of treatment by hydroxyurea, as shown here. So essentially, Jak2V617F, we know it induces DNA replication stress and can promote fork stalling, but in the normal scenario, we think that RECQL5 is upregulated to counteract this. But then when you knock out RECQL5, you basically exploit this dependency on RECQL5 and you start to see fork collapse and double-stranded breaks. And then when you put into the mix pharmacological inducers of replication stress, you really potentiate this effect and you start to see double-stranded breaks and development of apoptosis. Actually, we see an exon-positive cells, these cells undergo cell death. So in summary then, what I've shown you today is that RECQL5 is a downstream target of activated JAK2-PI3 kinase signaling. We found that RECQL5 functions specifically to protect Jak2V617F cells from both endogenous and exogenous replication stress by regulating fork stability, and that we can exploit this in the context of treatment with Hydrea to really preferentially target these cells, and there seems to be a synthetic lethal interaction between RECQL5 knockdown and Hydrea treatment in Jak2V617F cells. So finally, just want to acknowledge this work was led by Edwin Chen, who spent several years with me in the lab and now has his own lab in Leeds. We worked with Tony Green initially on these primary samples, and David Sykes and Dan D'Angelo I already acknowledge. And then several other members of the lab also contributed, and also want to acknowledge the people who, the foundations who funded this work. So thank you so much.